I also want to give a final review for the whole course. I really want to congratulate the course students. You have learned so much. Thinking about when you started this semester, what are the things that we looked at in this course? There are many different biological questions we evaluated, right? So transcription, how to look at transcriptomics data, understand transcription regulation and how transcription factors are regulating the expression of genes. We looked at also epigenetics, different epigenetic marks, different epigenetic status, how chromatin factors influence the reading, writing, and erasing of the different histone marks and nucleosome positions, right? We look at how to use epigenetics to understand GWAS. We talk about GWAS, EQTL, and how to use functional genomics data to understand the GWAS hits. And then we talk about cancer genomics and precision medicine, targeted therapy, and cancer immunology, immunotherapy. So these are all really kind of wide array of biological questions. But you also learn a lot of genomic techniques in this course, right? So we started from RNA-seq, later on also single-cell RNA-seq. For transcription regulation, we look at ChIP-seq. For epigenetics, we talk about bisulfite sequencing, DNase or ATAC-seq sequencing, single-cell ATAC-seq, and also Hi-C for chromatin interactions. For GWAS, we talk about case control studies or family-based association studies, also EQTL studies. For genome sequencing, we talk about germline sequencing, which is mostly for GWAS related, and also somatic sequencing, which is usually tumor and blood comparisons to look at cancer mutations. For screening, we talk about genetic screens and chemical screens. We didn't specifically go over these, but you can see a lot of these genomic techniques are being combined. For example, CRISPR screens are followed with single-cell RNA-seq, or you see combined single-cell RNA-seq with single-cell ATAC-seq. And recently, there are also single-cell RNA-seq with proteomics. And also recently, there is also imaging or spatial transcriptomics. So you can really see genomics adding to other genomics or other omics assays together. And so I think things are getting more and more complicated, but also more and more powerful. So computational biology will become more and more important. We covered a number of statistics and computer science topics as well. You have to learn a lot of R, bioconductor, and Python programming in this course. We talk about quantile normalization, how to test whether something follow a normal distribution using QQ-plot. We talk about a lot of statistical tests, t-test, chi-square test, Kamenov-Smirnov test for gene set enrichment analysis, Mann-Whitney U test for non-parametric test. This is for single-cell differential expression analysis. We talk about multiple hypothesis testing. Because in genomics, there are so many tests you have to do, we really need to care about correcting for multiple hypothesis testing, either using Bonferroni, which is very conservative, or false discovery rate, which is more popular in genomic assays. We talk about hierarchical models. This is kind of like RNA-seq. You are trying to borrow information from nearby genes to help you estimate parameters on an individual gene when you don't have so many replicates, right? We talk about generalized linear models, which is actually used in both LEMA for differential expression analysis or MAGIC for identifying differential guide abundance or differential gene selection in CRISPR screens. For clustering, we talk about k-means, hierarchical clustering, PCA, and later on in single cell, we also talk about using t-SNE and UMAP or graph-based network approach to kind of cluster the samples or visualize those cell populations or subpopulations, and also using the canonical correlation analysis to correct for batch effect. these are kind of the CS or machine learning approaches that people are using now in bioinformatics. We have discussed some machine learning approaches such as a support vector machine, random forest, neural network, lasso, and logistic regression approaches. And you've tried that in the homework as well. For sequence analysis for alignment, we talk about Boros-Waller alignment and also suffix array approaches for doing alignment. In motif analysis, we talk about expectation maximization and Gibbs sampler, and you can see how they kind of enrich on the different sequence patterns. We also talk about hidden Markov model and dynamic programming and how to use those to identify cell state, to identify TAD domains, CPG islands, and the copy number changes and things like that. And also we talk about survival analysis, right? And finally, we touched on many, many different bioinformatics algorithms. For next generation sequencing, we talk about FastQC and the BWA for read alignment. For RNA-seq, we talk about RNA-seq QC, which is kind of giving you the quality control, STAR for read mapping, SALMON for kind of pseudo alignment, and DE-seq for differential expression. For batch effect removal, you can either use Combat or Lima. And for gene ontology, we talk about David and gene set enrichment analysis. And for single cell, we mostly focus on Surratt, even though there are also many, many other analysis methods. For ChIP-seq, DNase or ATAC-seq, we talk about MAX and Cystron. And single cell ATAC-seq, you have some exposure on Maestro followed by some Surratt functions. And for genome sequencing, I guess we didn't specifically tell you how to run MuTech because it's a pretty time-consuming effort, but at least you can look at the MuTech result. And we showed you how to do CRISPR screen analysis and also how to look at some immunology analysis methods. Oh, I forgot, actually, in this year, you also have homework really looking at how to do DNA methylation analysis and also Hi-C analysis. So I would say we have covered a lot of algorithms. And I hope through this, I know a lot of you guys worked really, really hard on the homework. And I hope you can see bioinformatics, we have no way of teaching you all the possible algorithms, but at least you can see you don't need to be afraid of learning another bioinformatics tool or looking at a new data set. You can have the confidence to say, okay, I'm just gonna read the README, try to install the software and try to start a run and evaluate the result and troubleshoot the results and understand what to do next. So that's what I'm hoping the course will teach the students. All these techniques are developed because there are new data types or new experimental techniques that have computational or statistical challenges. And so informatics algorithms are developed, some for QC, some for exploratory analysis, some for batch effect removal, some for comparison, some for integration, and some for kind of downstream more biological insights. But in general, I think through the course in all the homework, we try to make each homework as coherent as possible so you can see how people think about a biological question. Okay, so yeah, I really kind of want to congratulate the course on the students on working so hard this semester to learn so many contents and hopefully give you the confidence to go forward as the field continue to progress.